Hello, my name is Jose Longoria, and on behalf of my colleagues, I will present this poster. We have no actual or potential conflicts of interest to disclose. Metformin is an oral antidiabetic agent with potential antineoplastic activity, which may be exerted through a systemic effect on circulating insulin levels, through modulation of intracellular growth and proliferation pathways, or both. However, its efficacy in clinical trials has been mixed. Our aim for this work was to explore how metformin interacts with cytotoxic agents in primary breast cancer cell cultures and triple-negative breast cancer cell lines. Briefly, primary breast cancer cell cultures and established cell lines were treated with increasing concentrations of metformin, paclitaxel, and carboplatin to estimate the EC50 for each drug alone, and combined with metformin with ultraviolet assays and nonlinear regression. The pharmacological interaction between the drugs, either synergism, antagonism, or addition, was estimated with the Chow-Tolai method for drug combination studies. Cells treated with paclitaxel and metformin at EC50 concentrations were lysed for Western blot analysis of anti-apoptotic proteins. All results represent at least 3 independent experiments. We used 4 primary breast cancer cell cultures, which appear in boldface on the table to the left, and 2 triple-negative cell lines, one of which is deficient in BRCA1. These cell lines have been fully classified by their epithelial mesenchymal transition phenotype, or EMT. This is relevant since our lab has previously described that metformin may revert an experimental induced EMT. The table to the right shows the doses used for cytotoxicity assays. Plotted on the left are the dose-response cell death curves for metformin. The vertical line number represents the EC50 for each cell line, and EC50s in the standard order of the mean for each cell line is plotted to the right. Broadly, cells with a mesenchymal phenotype are more resistant to metformin than cells with an epithelial phenotype. And in these plots, we can actually see 2 regression curves. The rightmost curve represents the dose-response curve for paclitaxel alone, and the leftmost curve represents the dose-response relationship for paclitaxel combined with metformin. In boldface, we can see the EC50 for the combination of paclitaxel and metformin, and in regular type, the EC50 for paclitaxel alone, which can also be seen in this plot to the right. The magnitude of EC50 displacement was generally higher in cell cultures with an epithelial phenotype. Combination index plots are shown in this slide. The x-axis represents the fraction of affected, which represents percent cell death, and the y-axis represents the combination index. A combination index less than 1, which can be seen in most cells except MBCDF4, represents a synergistic effect for the treatment combination. Combination index of more than 1 represents an antagonistic effect, and combination index of 1 represents an additive effect. In this slide, we can see the results of Western blot protein analysis after 24 hours of treatment with paclitaxel, metformin, or both at their EC50 concentrations for each cell line. Western blot protein analysis showed activation of VAX or caspase-3 in the epithelial cell lines, which were the most sensitive to metformin, although exposure to metformin didn't seem to modulate protein quantity for each cell across conditions. And in this slide, we can see the results for co-treatment with carboplatin and metformin. A luminal primary culture, the triple-negative cell lines exhibited the greatest displacement of the dose effect curve. The plot to the right shows that mesenchymal cells have greater resistance to carboplatin than epithelial cells. And as anticipated by previous plots, the triple-negative cell lines and the luminal primary breast culture exhibited the most synergy for co-treatment with carboplatin and metformin. Notably, HCC 1937, a cell line deficient with BRCA1, showed a synergistic interaction with co-treatment across all drug concentrations. This was expected, as carboplatin has been shown in vitro and in vivo to be more potent in BRCA1 deficient cells through synthetic lethality. Interaction between metformin and cytotoxic agents is variable between different drugs and cell lines. Paclitaxel combinations were synergistic in most primary cultures and cell lines, while carboplatin combinations were synergistic in a BRCA1 deficient cell line. Mesenchymal cells showed high levels of anti-apoptotic protein expression. However, metformin does not impact anti-apoptotic protein expression significantly. This begs the question for the potential for efficacy of BH3 mimetics in breast cancer. Thank you very much.